Hello everybody and welcome back to Creation Myths. Today's creation myth is common design. What are we talking about when we're talking about common design? Before we get into the actual creationist argument, we have to talk about a related term, and that term is homology. Homology is similarity due to common descent. It could be morphological, developmental, genetic, whatever. It's similarity in some characteristics of different organisms because they share a common ancestor. Similarity across diverse lineages is extremely strong evidence for universal common ancestry. Now, creationists will often call this claim, this line of evidence for common ancestry, circular. And that's wrong. Why is that wrong? Because based on homology, the foundation, the concept of homology, we can make accurate predictions about similarities and differences between different groups of organisms. The idea kind of operates at two different levels, right? So we can look at organisms that are similar in some way, right? We can look at the limb of a human and the wing of a bird and the fin of a whale, and we could say, okay, they're similar, they are homologous, they share a common ancestry. But for our purposes, in terms of being strong evidence for universal common ancestry, what we're really talking about with homology are the accurate predictions we can make. We can say, for example, okay, we can watch how limbs develop in one group of organisms and how they develop in a completely different group of organisms, like hypothetically flies and mice, and we can predict that those different limbs, which don't share a common ancestor with a limb, but we can predict that those different processes of limb development are going to be governed by and regulated by the same sets of genes that are homologous to each other, because even though the limbs are absent in the common ancestor between those organisms, the genes should have been present. And we can predict that we're going to find the same gene networks in those organisms. And that's exactly what we find. So here, for example, is the pathway. It's simplified, but this is the pathway governing the elongation of the fly leg. Here is the equivalent pathway for the mouse leg, and you could just kind of extrapolate these to arthropods and mammals kind of more generally. And you can see that the regulatory networks are the same, and if you compare these genes, for instance, HH right here and SHH right here, you're going to find a high degree of sequence similarity, right? Those are homologous genes, and they do equivalent functions in these pathways. That is a prediction that flows from the hypothesis of universal common ancestry. Now, the really cool thing you can do here is actually move these genes back and forth. So you could, for example, the experiment that I'm familiar with involves taking a fly gene that has to do with, I think it was limb development, and putting it in a bird embryo, a chicken embryo, and it'll actually do the same thing in that chicken embryo, which is super cool. This isn't just for limbs, though. We can look at the same kind of thing for the dorsoventral patterning during development. So this is looking at two major groups of animals, the protostomes and the deuterostomes. And dorsoventral patterning is back is dorsal and belly is ventral. And protostomes and deuterostomes are kind of like upside down from each other in terms of where like the nerve cord and the digestive system go. Turns out the reason for that is that the expression pattern of some developmental networks are flipped. So what's on top in the arthropod, in the protostome, is actually on the bottom in the deuterostome, right? So that's really cool. And then another great example of this is, of course, eyes. All eyes, whether they're in flies or mammals or cephalopods, or even like the very simple eyes that you find in things like cnidarians, all of these eyes are based on the same group of proteins to detect light. These proteins are called opsins. And even though the common ancestor of all the stuff with eyes definitely didn't have eyes, it had opsins, and it had the ancestral protein to opsin. So we could actually trace the evolutionary history of opsins through all these different groups. The important thing about this is that this is a confirmed prediction that flows from the idea that homology exists. If homology exists among different eyes at the genetic level, we should be able to find structures of similarity and common ancestry in the genes that govern their development and are used in their function. And that's exactly what we see. That's what makes homology such a powerful argument for universal common ancestry. So the creationist argument in response to all of this is, that's just common design. Whenever an engineer is designing something, they're going to reuse parts and modify them for different machines. It's the same thing with biological systems. The similarity is due to the same creator reusing parts rather than common ancestry. This supposedly explains the nested hierarchical structure that we find when we compare these systems across very diverse groups. So you can find this here from ICR. Is it common design or common descent? This is from 2018. This is actually a podcast episode. We have a critique of a review of an intelligent design book from the Journal of Creation in 2009. The author of this critique wrote, the author of the book that's they're reviewing, he also does not address the argument that similarity of structures could just as easily come from a common designer as from common descent. So that's the creationist argument. It's not common descent that explains this pattern, it's a common designer. Well, here's why creationists are wrong. These two models make distinct predictions, very specific predictions that are at odds with each other. So we're going to go through and we're going to see what the specific predictions are, and then when they've been tested, because they've been tested, we're going to see which model comes out on top. This is how we evaluate good science from bad science. Good hypotheses, strong hypotheses from weak, unsupported hypotheses. We see what predictions we make, and then we test them. That's what we do on this channel. We're going to take creation science seriously, we're going to treat it as legit science, and we're going to see exactly why it fails. So what we're going to do here is we're going to look at the common design model and the common ancestry model. The common ancestry model can be treated as one thing because the entire genome, right, shares common ancestry for everything, or at least all cellular life, according to that model. Now, within common design, we have to split it into two groups. We need to look within the created groups, or sometimes you hear that as created kinds, right? So we have to look within the kinds, because those groups, those organisms, share common ancestry. And we also have to look between the created kinds, because those different groups do not share common ancestry. Within each of these, we're going to look at constrained sequences, that's sequences that are constrained at the nucleotide level, like the sequence of the bases actually matters. And then we'll also look at unconstrained sequences, sequence where the actual sequence of A's, T's, C's, and G's doesn't actually matter for whatever reason, right? So let's look at what we expect within these different models. Now, in terms of constrained sequences, it's easy. These are sequences that have some functional constraint. So across the board, we expect nested hierarchies. For common ancestry, of course, we expect that because they share a common ancestor. For common design, whether they're within or between created groups, we expect nested hierarchies because they share a common designer, right? So even between different groups, if you're making eyes, you're using opsons, we expect the opsons to recover a nested hierarchy. But way more interesting are the unconstrained sequences. Now, for common ancestry, we expect the unconstrained sequences, the sequences where the actual order of the bases doesn't matter, and you can have different bases and it works fine. For common ancestry, of course, we expect a nested hierarchy. Why? Because everything shares common ancestry. For common design within created groups, we also expect a nested hierarchy, because within the groups, you share common ancestry. But between the created groups, where we don't have constraint at the sequence level, and we don't have common ancestry, there we expect those unconstrained sites to be uncorrelated between those groups. Now, I want to dig in a little more into this to make sure we're all on the same page before we test these predictions. So why are these our specific predictions? Well, the creation model that we're working with here could be called the orchard of life, as opposed to the tree of life for universal common ancestry. And the question we want to address is, what do we expect in the unconstrained regions? This figure from AIG is going to help us explain. What's going on here is we have the original created kinds, and then we have their evolutionary history from creation down here to present up here. This little level in here indicates the flood, but you can just ignore that. It doesn't matter for what we're talking about today. So what we want to look at is within the created kinds, right? If you have regions that are not constrained, the actual letter of the bases doesn't matter, right? Within those sites, within a created kind, we expect the mutations that we see to be correlated, right? The mutations in this lineage should be correlated in some way to the ones in this lineage and this lineage, because at some point in their history, they share common ancestry. But if we were to compare the mutations in the unconstrained sites across different created kinds, they should be uncorrelated with each other, right? The mutations that have occurred in this lineage have nothing to do with the mutations that have occurred in this lineage because they don't share common ancestry. So we can now test these predictions and see, do we see correlated within the kind but uncorrelated between them? Or, as we expect with universal common ancestry, do we expect a nested hierarchy across kinds? That's what we're able to evaluate. So what do we see? Well, we find nested hierarchies in constrained and unconstrained regions. I'm not going to show you any data for the constrained regions because everybody agrees on that. Everyone's predictions agree with each other. But let's look at these unconstrained regions. We can look, for example, at regions called ERVs, endogenous retroviruses. That's ERV. Those are viruses that integrated into a genome at some point and then broke, and now they're just kind of stuck in there. And we know from looking at lots of different genomes that ERVs are unconstrained. They accumulate mutations at a rapid rate, indicating that the sequence of the ERV is not constrained. And in fact, most ERVs are largely degenerate. Much of them is actually been lost over evolutionary time. What do we find when we look at ERVs? Well, we can reconstruct, for example, primate phylogenies based on their ERV sequences. We can also look, that's one paper from 1999. In another, we can look at how we can recover distinct groups of mammals like simians and rodents, and they fall out separately. Now, creationists would clearly call them different kinds, different created kinds. So there's no reason you should get this nice nested hierarchy if they didn't share common ancestry according to the creation model. You can also look at sites within genes that are not constrained. These are called wobble sites, and there's different hypotheses about the degree to which wobble sites are limited by selection in terms of their variation, but in general, selection acting on wobble sites is extremely weak. In this study from 2016, the authors tested for a signal of common ancestry in these wobble sites. So again, these sites where it's the same amino acid, but it's a different nucleotide. What they found, and I'm just going to read what they wrote because they tested a bunch of different models for the degree to which these sites are constrained and what the null hypothesis is in terms of their variation. I'm just going to read what they wrote. They said, across all 6 conditions, the observed data had much lower entropy than expected under the family SA hypothesis. That's family separate ancestry. And that's useful for us because creationists often put those common ancestors for the created kinds at the family level, right? So each family is representative of a created kind. The authors continue, these z-scores correspond to p-values effectively equal to 0 and represent enormously strong rejection of the family SA model. Again, enormously strong rejection of the family separate ancestry model. So what we have here are separate predictions from the common design versus common descent hypotheses, and we have here direct refutation of the common design hypothesis by showing that its predictions are not just wrong, but are completely wrong. They are the opposite of what we actually find. So what are the creationist responses to these findings? Well, you find them in 2 flavors. First, you get, God could have done it that way, which, I mean, yeah, sure. In theory, that's true, but that's unfalsifiable special pleading. Thanks for playing. What's next? The more sophisticated response is that, well, all DNA is functional, so we should always find nested hierarchies. Now, this claim is based on the findings reported from the ENCODE project, which was documenting biochemically active and functional parts of the human genome, and it's not true. This claim right here, that all DNA, at least all the human genome, is functional, is not true. For a clear refutation of this, you should see the 2014 paper defining functional DNA elements in the human genome. This is the follow-up paper from the ENCODE team to their very provocative and I will say over-the-top 2012 paper that was horrifically overinterpreted as, the death of junk DNA and silliness by tons of people. Creationists love to glom onto that 2012 paper, but they ignore the 2014 paper that was a little more circumspect about their findings and very clear that a lot of the biochemical activity they're reporting is actually not indicative of function and is actually specifically indicative of non-functional sequences. But let's put that aside. I will say I did a video on that. I'll link it so you can check that out. I've talked specifically about that claim. But let's put that aside. Let's assume that they're right. All DNA is functional, so we should always find nested hierarchies. Even if that was true, all DNA being functional, some functional DNA is not sequence constrained. Right? You've got spacer DNA where the length matters, but the sequence doesn't. That's functional. You need that DNA, but it's the number of bases, but not the specific sequence of bases matters. In other parts of the genome, it's not the specific bases, but you need a certain amount of G or C, right? But it doesn't matter which ones are which as long as you cross a certain threshold. And then we actually just looked at some functional but non-constrained DNA when we looked at the wobble sites. That's actually within protein coding genes. So it's certainly functional, but it's not sequence constrained. So even if this claim that all DNA is functional is true, it's not. But even if it was, we still find violations of the creationist prediction. So in summary, creationists claim that common design explains the nested hierarchical organization of biological systems. But conveniently, common design and common ancestry hypotheses make distinct predictions. And we can test those predictions, which has been done, and the results are in line with common descent and definitively refute common design. That is, of course, unless creationists want to concede defeat and resort to special pleading, but in doing so, they make their argument non-scientific. So, common design as an explanation for the structure of biological diversity, that is a creation myth. Thank you for watching. Please like, share, and subscribe. See you next time. Don't get fooled.